Okay, so I think we will go ahead, and as people start joining, I will allow them into the call. But that being said, I want to welcome everyone to the call. My name is Gabriel. I am with Malmberg Capital. And today we are hosting a presentation with Dr. Satish and his team. Now, he's the founder and CEO of Immgenuity, and they are based out of the United States. Now, Immgenuity is a life science company, and they are at the absolute forefront of HIV treatment innovation, and they are creating a therapeutic vaccine to treat HIV-associated neurological inflammation and cognitive disorder. And these are areas that are not effectively addressed by antiretroviral drugs. Now, the company's product aims to restore natural immunity and reduce the inflammation in the central nervous system. This cutting-edge approach also addresses HIV drug resistance, and they're now preparing to submit their IND to regulators and plan to complete initial manufacturing and phase 1 trials 20 to 30 months after funding. Hopefully, I got this correct, Satish. The company is seeking $30 million for their first funding round to facilitate technology absorption, production, and of course, the clinical trial preparation. So that is let's say, the short introduction into Immgenuity. And feel free to write questions in the chat as they come up, and we will answer them either throughout the presentation or we will just hold a Q&A at the end. Of course, I will share this presentation with you at the end of the call. So with that being said, Satish, I'll pass it over to you, and if you want to begin by giving yourself a bit of an introduction.
Bine mulțumesc. Thank you. Thank you for the opportunity to give this presentation. My name is Satish Apte, as he explained, and I am a neurologist and an HIV researcher for over 20 years. And to give a brief background on me, I graduated from medical school, then did a research fellowship at Hafkin Institute in Mumbai, and then also at l'Institut Pasteur in Paris, where I worked in encephalitic viruses. And there I caught the eye of a professor from Harvard who invited me to continue my training at Harvard. So I ended up in Harvard Medical School. And I finished my neurology residency and a clinical fellowship at what used to be Peter Bent Brigham Hospital. Now it's called Brigham and Women's Hospital. It tells you about my age. And then I was at a meeting of American Academy of Neurology and met a professor from Stanford, and he wanted me to go to Stanford to continue my fellowship. So I did a fellowship in neurotransmitters and neurology in Stanford. And then I was in private practice for a short while until I came up with the idea of treating HIV patients. And that is where this invention arose. Let me introduce you to my team very quickly. We have Dr. Marcel Kerlin, who is a professor of infectious diseases at Oregon Health Sciences University and an HIV researcher. We have Dr. Richard Ogden, who is a former principal of Aguron Biopharmaceuticals, which brought, and he was in the team that brought one of the first protease inhibitors to market, and then shepherded the sale of Aguron to Pfizer. We have Mr. Nagy, who has over 30 years of experience in accounting in private and public companies. He's our CFO. He's a CPA in CGFM, which I don't know what that means, but sounds impressive. And then we have Dr. David Miller. And Dr. David Miller is an epidemiologist who has worked in HIV for over 35 years. And he was involved in a lot of task forces and councils. So he was on Mayor Michael Bloomberg's HIV Council, which had over $7 billion in funding. And he shepherded a lot of those programs. Dr. Miller is also on the other side as an investor. He was working at Beyond Capital in Netherlands, as well as Spencer and Trask in New York as a partner. And he has a lot of experience in investing in biotech and in HIV particularly. So, and he has attended over 200 scientific meetings about HIV, and knows a lot about HIV and how the government funding works, how the investor funding works, and where the gaps are in HIV treatment. So he is very valuable as he's our strategic advisor. So with that, I'll just give a brief elevator pitch on what we are trying to do. And what is happening in HIV right now is that we have a lot of antiretrovirals which are slowly transitioning to long-acting antiretrovirals, which are no improvement from the regular antiretrovirals in terms of actual pharmacodynamics, but they are long-acting, so people have to take them less frequently, and thus become more popular. But as the antiretrovirals suppress replication of HIV, they are unable to suppress the replication in what are called anatomical reservoirs, where low-grade viremia persists, even though the patients are taking the drugs faithfully and in their blood, their virus is undetectable. But even when they're well controlled, they still have this going on inside them. And that results in potentials for drug resistance, as we have seen with PrEP, pre-exposure prophylaxis. People who receive PrEP have a 30% chance of having drug resistance to common antiretrovirals. The other issue is that this low-grade viremia continues to have, there are two immune systems, basically adaptive and innate. And the innate immune system is primarily used to defend against bacteria, but it's also present everywhere in the periphery and is used as an alarm system. So when you see an offending organism, the innate immune system sounds alarms. And these alarms are called chemicals called cytokines. And they start circulating, which triggers the adaptive immune system to come and take care of the problem. So in HIV, the adaptive immune system is completely paralyzed by the HIV. And so the innate immune system keeps on sounding this alarm over and over and over, causing increasing cytokines in the blood. And they circulate all over the body. And these are highly inflammatory cytokines. And they create various disorders in the brain. They cause HAND, HIV-associated neurocognitive disorder. They cause various autoimmune conditions. They cause tumors, Kaposi sarcoma. HIV patients who are taking antiretrovirals faithfully still have a 17% higher rate of getting tumors. And also, it causes cardiovascular disease. So HIV patients are more likely to require coronary artery bypasses. And the same goes for effect on nerves, causing peripheral neuropathy, and also kidney and liver issues, because these cytokines are causing inflammation that is remaining there constantly. And this is in people who are taking the drugs faithfully and have their viremia well controlled. That means undetectable or below 20 copies per mil. And currently, the establishment has no solution to it. The medical community in HIV has no solution to this at all. And so after studying about 1,400 papers on this little gene called NEF, which has been labeled as an accessory gene, but it is probably the most important gene in the HIV virus. And this gene single-handedly stops almost 11 different ways in which the immune system can attack HIV. And so the theory is that if you remove the NEF gene, you'll render the virus not only non-pathogenic, but it will also train the immune system to attack it properly, because NEF will not be able to interfere in the immune response. And there is evidence of this in literature that people who have defective NEF genes either become elite controllers or they don't get sick at all. So this evidence is there, that hundreds of reported cases where a NEF defective gene and the degree to which that protection is given is directly correlated with the length of the defect in the NEF gene. So the longer the defect, the more likely that the patient will become an elite controller. And so with that said, there is an absolute need for this product. There is nothing like it. There's no competition. And it will be a very simple treatment that can be used worldwide. It will consist of just intradermal injections. And so I will go into what is in the pipeline from other people in my presentation. So with your permission, I would like to display the presentation. Can everybody see it?
Yes.
Yes.
Okay.
Yeah.
Okay. Let me. So this slide was created because I get asked all the time that we don't really need any treatment for HIV anymore because we have it under control and it's gone. So what is critical is that 71 patients die of HIV every hour in 2022, and that trend is continuing. And so it's not over, and some radical approach is needed in order to save publicly funded programs like PEPFAR and Global Fund. So this will lead to a remission and then a cure. And it's an immunotherapy. So we are going to disrupt the global antiretroviral market, which is valued at over $75 billion, and achieve durable clinical remission. And I won't just read from the presentation because it puts people to sleep. So I would rather just go with highlights. So it's a novel therapeutic vaccine. We have completed the CGMP and the required animal studies, and we are preparing for an IND submission to regulators. One of our SAB members is a former senior scientist at CBER in FDA, and he's going to be working on getting the submission for IND. And we plan to complete phase 1 trials within 24 to 30 months post-funding. And our initial study is expected to give us efficacy data and get fast-track designation. And subsequently, for low to middle income countries, we plan to license the technology or get acquired or conduct an IPO as exit strategy. So I just described what is out there. that they don't really have anything to address HIV the way it stands today. So there is you probably heard of this shock and kill approach, and they use what are called latency reversing agents. And so far they have failed in 24 different studies. And the issue is the same. If the antiretrovirals cannot reach the reservoirs, how are the latency reversing agents going to reach there? Then there is a block and lock strategy. So shock and kill means you force the reservoir cells to start replicating and then kill them. Block and lock is to prevent them from replicating to begin with and then lock them in place so that it never starts producing HIV virus again. And those have had massively disappointing results. Then there is a CRISPR-Cas9, where you take a gene editing tool, put it into an adenovirus vector or some other vector, and inject it into people. And the CRISPR hopefully goes and cuts all the HIV provirus from the latent cells. Most people think that this is not likely to happen because, first of all, you can give it only once, because once you're given adenovirus, the next time the body's immune system attacks it, and the CRISPR cannot go where it is destroyed before it can reach its target. And the second thing is that you'll have to use so much CRISPR, because each CRISPR Cas9 is consider that as a pair of scissors, and they go and cut the target of the DNA. And the problem is that once it cuts it, that thing is ineffective after that. So you'll have to give a lot of scissors. And there are over 10 billion cells in the HIV reservoir. And so you'll have to give 10 billion scissors, each one contained in an adenovirus vector. And that will become like 1 liter of injection to give. And it is unlikely that the FDA would allow that, and also it is unlikely that any patient would survive 1 liter of getting adenovirus. And then there is CAR T and gene therapy, which has to be administered individually. So it's tailor-made for each patient. So you take the patient's blood and train the T cells. CAR stands for chimeric antigen receptor, and tailor-make these T cells of the adaptive immune system and inject them back into the patients. This has had some success, especially in oncology. There haven't been any significant clinical trials in HIV patients, and even if it works, it's going to cost over $300,000 per patient, and several clinic visits, and it is not practical to give it in low to middle income countries around the world. So it's not a practical approach. So I've already explained the unmet medical need. So we are IND ready, and our focus is infectious diseases and immunology. And our vaccine will restore the adaptive immune system, which will in turn silence the innate immune system, which is sounding the alarm with cytokines. So it will silence the circulating cytokines. When the circulating cytokines are silenced, the brain, heart, kidney diseases and peripheral neuropathy will be also helped and resolved. And our scientific advisory board is extremely eminent. And I didn't bring all of them into this presentation. It would have been just overwhelming. But Dr. Dan Karitskas, who is the chairman of infectious diseases at Harvard, has over 650 publications in peer-reviewed journals. Dr. Carol Brogard is an clinical professor at UCSF, and she is the former director of medical research at Gilead. And she has an extremely extensive research history in the field of HIV. Dr. Jürgen Rockstroh, chairman of infectious diseases at University of Bonn, Germany. and he's a former president of the European AIDS Society. Dr. Liddy Trotman, who is an immunologist, and she's at the Military HIV Research Program, MHRP, and she's the lead in immunology and translational research. Dr. Ogden, who is present here, and I already briefly described him, that he is one of those people that has actually brought an HIV drug to market, from conception to market, through all the phases, and then successfully shepherded the sale of his company to Pfizer. Then Dr. Ron Sakura, that I talked about, he is a former NIH researcher, as well as a former senior scientist at CBER, Center for Biologics in FDA. And he will be helping us with all our FDA meetings, etc., and the submissions. We have Mr. Jesse Milan. He's the president and CEO of AIDS United, and he has been involved in reviews of a lot of research proposals, and he's on the scientific advisory board of PEPFAR, and he's also a member of PACHA, President's Advisory Council on HIV and AIDS. Dr. Jessica Robinson-Papp, she's a professor of neurology at Icahn School of Medicine, and she works in neuro-AIDS pathogenesis and treatment. Dr. Marcel Kerlin, who is here with us, and is a professor of infectious diseases at the Oregon Health and Sciences University, and is a leading HIV researcher and clinician with several publications. Dr. Roscoe Moore, who is a former Assistant Surgeon General of the US, and has been at the forefront of the HIV policies, including PEPFAR and MHRP. He has been instrumental in getting those things funded through Congress when he was in the position of Assistant Surgeon General. Dr. Satish Chandran is a former Senior Vice President of Vaccine Manufacturing at Wyeth and Pfizer. And so when we come to large-scale manufacturing, he would be extremely valuable. Dr. Linus Vanderchkov, $10 to anyone who can pronounce it. So he is the director of the HIV Cure Research Center and professor of infectious diseases at the University of Ghent. And he will be another principal investigator with Dr. Kerlin to do clinical trials. He's willing to do it. And then we have Dr. Wojciech Witkowski, who is a senior researcher at the HIV Cure Research Center in Ghent, in Belgium. And so it's a live attenuated therapeutic vaccine to treat HIV-infected patients to establish immunological control of HIV. And we have a scalable and practical manufacturing design capable of distribution and administration worldwide without serious side effects.
I'm going to take a second here to jump in. This is
Okay.
David Miller. The scientific advisory board you just saw, that's now subscribed to the premise Immgenuity is putting forward, that a therapeutic vaccine can bring about remission for HIV, indicates that within a year's time, this company has been able to solicit significant scientific agreement amongst the leaders of international clinical trial networks that are publicly sponsored. Every HIV drug that's come forward has been sponsored by federally facilitated clinical trial programs. You have a situation here where the leadership of those clinical trial networks is reflective and representative of this SAB. So that unto itself is somewhat of an extraordinary achievement of significant consideration. Because if we're going to go through clinical trials, and we're going to have to have that consensus amongst those leaders. I thought that might be important to identify for you.
Thank you, David. So the scientific rationale, I briefly touched upon it, but let me kind of go over it again. So NEF deficient HIV. Okay, so for those who are not in biotech, NEF in lowercase and italicized means the gene, and NEF upright and the first letter capitalized is the protein that gene makes. So NEF deficient HIV infection has been shown to lead to elite suppressors and long-term non-suppressors, long-term non-progressors of HIV infection. I talked about that. This is the clinical proof that NEF is responsible for the pathogenicity. It is postulated that the NEF protein renders HIV pathogenic by interfering with cytotoxic T lymphocyte signaling by using 11 different strategies to defeat the immune system, which directly protects the infected cells from attack by CTLs. And so giving IMTV-014, our vaccine, to already infected individuals harnesses the power of the CTLs, just as in rabies infection. In rabies, we give a post-infection vaccine. We don't go about giving prophylactic rabies vaccine to the whole neighborhood. We only give it to people who have been bit by a dog, fox, or bat, or something like that. So that same approach is what our vaccine does. And so it's likely to reverse the effects of HIV-1 NEF protein, restore immune signaling, and achieve immunological control, reduce cytokine production and neuroinflammation and astrocyte malfunction, reversing the progression of HAND, which is HAND stands for HIV-associated neurocognitive disorder. Now, this is another one of our achievements, which is spectacular. We have had 8 presentations in just 1 year. So we presented at HIV DART conference in Cabo San Lucas, then at XMID conference in Copenhagen in April, Italian conference on retrovirology, also called ICAR, in Bari, Italy in June, then US conference on HIV and AIDS, Washington, DC in September, HIV Cure Symposium in Ghent, Belgium, and then 5th World Infectious Disease Conference in London, and the conference, World Infectious Disease Conference in Sapporo, Japan. And so there was one conference where we were accepted to present. That was iSheet conference. Unfortunately, that conference got canceled. So we have been very successful, and we have been getting a lot of attention from these conferences. We have 3 collaborations.
I'm going to take the opportunity to jump in again,
Sure.
which Dr. Opti's gotten used to, my aberrant nature of doing this. But it's important to note, getting this many conference presentations in a year's time is unparalleled. What Opti and the rest of the team here has done is driven scientific consensus that NEF needs to be approached as a new target for not just HIV, but for a better understanding of retrovirology. And that has to be done with a therapeutic vaccine. These are peer-reviewed conferences. And to have that volume of presentations accepted and in a very, very, very competitive environment is extraordinary. This coming summer in Munich, we're going to have an oral presentation and possibly an invited symposium. At that point, this shifts a great deal because it goes from demonstrated scientific consensus to universal command of getting the studies we need sponsored by the NIH, by the EATN, and other investigational research networks.
Thank you, David. So we have 3 collaborations: Oregon Health and Science University, BioStacks, and HIV Cure Research Center in Ghent. We are working with AVAC and Gladstone Institutes and a few more institutes and universities for collaborations. One of them is Karolinska Institute and Pasteur Institute. So those places take a long time in their bureaucracy to get an agreement signed, but they have shown willingness. So I won't go into the mechanism of action because I talked about it twice already. So rationale for targeting the HIV NEF protein. So NEF is demonstrably implicated in immune escape. So HIV is evasion of the immune system, making alpha beta CTLs, which are the adaptive immune system, that anergic. And I can go into more detail about this, but that is really going too deep into the immunology forest. So I'll keep it on the surface. NEF also promotes infectivity, promotes inflammatory cytokines from the gamma delta cells, which are the innate system cells that I talked about, disruption of autophagy in immune system cells, promotion of bystander cell death. Bystander cell death is a unique phenomenon that cells that are not even infected with HIV tend to die. And they're called bystander cells. And so I can go into more details about how that works. And then contributing to HIV persistence by disrupting 11 different signaling mechanisms of the immune system. So we have established that HIV NEF is the primary driver of HIV persistence, immune escape, and comorbidities. And like I talked before, over 1,400 studies in peer-reviewed literature established these three facts. Imgenuity's IMTV-014 is the only developing therapy targeting the actions of NEF. There is no one else doing this. Again, I won't go into too many details of this, but we have completed the preclinical toxicology study in mice, in rhesus macaques, in infected rhesus macaques, and humanized mice. And the current strategy to move forward from bench to bedside. So we are going to continue to build consensus and support from major institutions. We are also working with a GR firm to get support from congressmen. We have had discussions with Congressman Adriano Espaillat, and then Congresswoman Paulina Luna of Florida, Victoria Spards of Indiana, and Barbara Lee of California. And they all sit on the Congressional HIV Caucus. And we are trying to get support and perhaps a targeted line item funding for this project from Congress. We have established a dialogue with the Presidential Advisory Council for HIV and AIDS, Ms. Marlene McNeese, who is the co-director of PACHA, and our own SAB member, Mr. J.C. Milan. Through them, we are trying to create an awareness at the Presidential Advisory Council level for this vaccine. And then, let me move this away. And so we have developed a validated strategy for obtaining an IND from FDA to begin a phase 1 clinical trial at OHSU and perhaps University of Ghent. And our phase 1 trial will be open label. It will not require a double-blind control because we are going to show and demonstrate a remission after getting this vaccine, a drug-free remission for a period of 24 weeks. If we show that in 5 to 10 patients, it is something that has never been done in HIV before with a therapeutic vaccine. Our management team consists of myself, Mr. Negi, who is here on the call. It's pronounced Naj in Hungarian, but in South Carolina it becomes Negi.
Actually, in Hungarian, it's nudge.
And then we have Mr. Kevin Phelps, who is a director. He's also a CPA. So I don't know if having two CPAs is better than one, but he was a COO at a Kodak subsidiary, and then he has an extensive experience in biotech companies. Then we have Major General retired Kevin Leonard. He's got a lot of corporate management experience as CEO, COO, and board member at various companies. We have Mr. Dean Petkanas, who is the CEO of another company, and he has a lot of experience in drug development as well as in financial markets, public markets. And we have Senator George Allen, who is former senator of Virginia, former U.S. senator from Virginia, former governor of Virginia, brings a lot of government and management experience to create a splendid corporate governance at Immgenuity. And then we have Mr. William DiCandido, who is a counsel, in-house counsel. And actually, Dr. Richard Ogden is also on the board of directors, but this I didn't want to create another page for this. But Dr. Richard Ogden sits on both the scientific advisory board as well as on the board of directors. So patents and patent strategy. There have been a lot of patents that were granted that expired, that two that are still valid until March 28th, 2024. But that's not a cause for alarm, because we are going to be applying for additional patents, over 50 to be exact, because there are a lot of parts of our manufacturing process that have not been patented, and we have innovated them. So we are just waiting for meeting the requirements for 35 CFR 112.1. As soon as we finish our clinical trials, the phase 1 trial, we will be able to publish, we'll be able to apply for additional patents, both in the US and internationally. So there is a lot of technology that is patentable but hasn't been patented. Now, people in the biotech industry, they already know how much money the drug companies make overall. So business basics, revenue stream, like most pharma products, it will generate revenue stream from distributors in the developed world and licensing in emerging markets. Pricing will be based on per injection. Different pricing models will be utilized in developed. This is very standard pharma strategy. Customers, our end customers, are entirely people infected with HIV. The statistics of market size developed by the WHO and UNAIDS, they will get the vaccine via doctors and clinics. Sales strategy, being a regulated product, IMTV-014 will be sold only to physicians, clinics, and hospitals via established medical distributors. Distribution channels are also standard in the pharma industry. McKesson, AmerisourceBergen, Cardinal Health, and for foreign distribution, it will be PEPFAR and Global Fund. They purchase and distribute HIV drugs in developing countries, and they pay for them too through the PEPFAR program. Positioning. So it will be positioned as the final treatment for HIV, causing remission, leading to a cure. So I don't think it's common sense that people would want it much better than taking the drugs till the end of time. And it will be marketed primarily to physicians via scientific meetings, peer-reviewed journal articles, and advertisements in medical journals and HIV-specific media. So that is the marketing and PR part of this. Timeline. So first year, we're going to be doing the GMP manufacturing, site audits, protocol development, and then IND submission. And year 2, we'll have IND approval, and then phase 1 clinical trial at OHSU. So it depends upon if the IND approval takes 6 months or 3 months or 2 months. Hopefully we'll be able to get it in 3 months, but then we can go straight into phase 1 clinical trial. So in year 3, we'll complete phase 1 and get fast track designation and begin a phase 2B/3 trial commencement. To get a fast track definition, you don't have to do all the different steps in the thing. In year 4, we'll complete phase 2B and phase 3 completion, FDA application and approval process. And then in year 5, we'll start commercial sales. In year 6, we'll be doing licensing, manufacturing, approvals and sales in emerging markets and other countries. So usually if we get US FDA approval, then we get a shorter path in European countries and in Japan, in Coast Asia, and in Australia. So pricing analysis, I won't go into too much detail on this, but in the developed world, which has 6.2 million HIV positive people, HIV costs, treatment costs between $18,000 and $160,000 per year. So the $160,000 is when they are on BNABs, broadly neutralizing antibodies, and the new long-acting antiretrovirals, Cabenuva and lenacapavir. They cost a lot of money. In the emerging markets, the HIV treatment costs between $2,000 and $4,000 annually. The emerging markets are like Brazil, India, Thailand, etc. In the undeveloped world, the costs are between $1,000 and $1,500 annually, and these are entirely paid for by Western programs, PEPFAR and Global Fund. And the costs that I'm describing here, they include drugs, lab monitoring, clinic charges, and physician visits. So, so for a quick market acceptance and rapid distribution, we have strategized to position IMTV-014 competitively. This is all speculative. We don't know what will happen by the time we come to that point, but at the moment, for the sake of forecasting, in the developed world, the complete therapeutic vaccination will be priced at $10,000, and it is needed only once. So insurance companies would welcome that. Instead of paying $30,000 to $100,000 every single year till the end of time, they would love to pay the $10,000 right away, and just one time. In the emerging markets, the same. They'll be priced at $2,000, and in the developing world at $1,000. So to remain competitive. And then we made assumptions of how many people are living with HIV. The source is from WHO and UNAIDS. So there are 6.2 million in the developed world, 11.4 million in emerging markets, and 21,400,000 in the developing markets, a total of about 39 million people. So the current annual cost of treatment, which is obtained from CDC and NIH. And so pricing assumptions, number of doses is 2, developed 10,000, emerging 2,000, developing 1,000, and market share. So we will not be marketing until year 4. So in year 4, we assume we'll get a market share of 5%. In year 5, market share of 15%. In year 6, market share of 25%. And then there's a distribution and retail percentage that will be deducted from those prices that I mentioned above. We assume them to be 40%. And so cost of goods sold per dose is about $200, and overhead percentage of gross is 22%. And with those assumptions in mind, and we kept them very conservative, the distribution cost is, and retail cost is less than 40%, but for the sake of having realistic numbers, we have used those assumptions. And so you can see that just multiplying by the 5% market share and 6.2 million people, multiplying that by 10,000, you get these numbers. And so estimated EBITDA in year 5 will be 1,962,000,000. In year 6 will be 5,887,000,000, and year 7, 9,811,800. So large numbers, and but they have been brought up from significant and very conservative assumptions in the slide. When you see the presentation, you'll see the numbers. So estimations of EBITDA per share, we are again assuming that there will be 30 million shares outstanding and 35 million in year 3. And so investment of 30 million, we need another 20 million in year 3. And so when you divide the EBITDA by the number of shares, you have $56.07, $168.20, and $280.34 per share. So we need $30 million in 2 tranches. We wouldn't mind in 1 tranche, but and the use of proceeds is corporate infrastructure creation, recruitment of the staff, process of manufacturing the vaccine at a BSL-3 facility, release testing and certification of the new vaccine batches in preparation for phase 1 trials, first 3 years general working capital, including regulatory filing expenses and NIH grant development costs, and membership in bio and other conferences. And then complete technology absorption, preparatory work to enter phase 1 clinical trials via NIAID and IMH, and phase 1 study costs. This again structure in 2 tranches, 20 million in the first tranche. The second tranche will be due as soon as we have IND from the FDA. And that those 10 million will be spent for the clinical trial itself. And that is the end of the presentation. Thank you. And I will await any questions.
Thank you, Satish, for all of the. I mean, realistically, for those that wealth of details. So we'll wait to see what questions might come up in the chat. In the meantime,
Gabriel,
yes.
Sorry for interrupting you, but since Dr. Kurlin is busy and he has a clinic to attend, I would respectfully request that if anybody has questions regarding the clinical trial and his expertise in it and how he plans to conduct it, those be asked first, and so that he can leave.
Okay. So, a question that I have here that I think might have to do with what you just pinpointed out, Satish. So, can you elaborate on the collaborations that Immgenuity has established with other institutions? That would be one. And the other one, if you could provide additional details on the results of the preclinical toxicity tests and tests in humanized mice and macaques.
Yes. The first question is about the collaborations with OHSU and with University of Ghent. Those are the most important. BioStacks is a commercial company, and that is, we're going to be doing a collaborative research with their low-dose naltrexone product that enhances the immune response and reduces inflammation. But the main important ones right now are the OHSU and University of Ghent, the HIV Cure Research Center. And these are both extremely well-equipped and respected research institutions which have HIV patients, they have HIV clinics, they have HIV programs, and they will be able to undertake a clinical trial. They have also fully functioning laboratories and clinicians like Dr. Marcel Kerlin and Dr. Linus Vanderchkov. They're both well-respected and experienced researchers. And so those clinical associations will be very, very valuable. We are working on others that are more in the realm of advice and guidance so that they can give us advice and guidance on actual protocols, reviewing them and finding pitfalls and also making the protocols pass the IRB. The second question was about the preclinical toxicology studies. Now, these toxicology studies are primarily done for safety, and it's a standard model that we have with the FDA. So you do the mice and the rhesus macaques. We went one step further and we did infected rhesus macaques as well, because some people may have a question that what if a macaque, what if someone is immune compromised? Can they handle this vaccine? And so we did infected rhesus macaques and showed no toxic effects from this vaccine. And then we did humanized mice. And that is to, again, show that it is safe in humanized mice. Unfortunately, there is no animal model to test this vaccine for efficacy. There isn't one. And because SIV and HIV are completely different viruses and they operate differently. So you cannot, in HIV, we can cut the NEF gene from the beginning to 276 base pairs. You cannot do that in SIV because that gene overlaps with a couple of very important genes. And so if you try to cut it from the beginning, the virus dies. And so you can only start cutting at about base pair 82 and then cut it for about 100 base pairs. That's the maximum you can do in SIV. So there is no animal model for testing or similar vaccine in animals. And if we try to do in BLT mice, for those who don't know, are humanized. They grow with human immune systems by a transplant of bone marrow, thymus, and liver. And so they are just like humans, except they don't have lymph nodes, unfortunately. So you cannot measure reservoirs in BLT mice. But the problem is that they're irradiated right at birth to kill their mouse immune system, and then they're given a human immune system. So their life expectancy is at the most 4 to 6 months. And so by the time you give them HIV and have a set point, 2 months go by, and then there is not enough time to give 2 injections of the therapeutic vaccine to see if they get better or not, because by that time their natural lifespan approaches and they die. So there is no animal model right now for testing our vaccine. But the toxicology studies, none of them showed any toxicity from our vaccine in any of those models.
Okay, understood. I think this question actually came right on time because there was another question that said if you have any preliminary information on the existing studies. So I think that was just asked. But again, from the same person, Have you received any grants, and if so, what is the amount of the grants that you received up until this moment?
Yeah, we haven't received any grants until now because our company is very, very new. And but we will be applying for grants as soon as we are funded and staffed. And we are fairly confident that we'll be able to get grants through the NIH.
Okay. So what another question that we have, what specific milestones and timelines are associated with the 30 million funding round?
Excuse me, can you please repeat that?
Yes, what specific milestones and timelines are associated with the $30 million funding round?
Yes. Once we have the $20 million, we'll go into action to move forward with the BSL-3 facility and manufacturing three batches. FDA requires 3 different batches to evaluate for the audit. And so we'll get that audited. Our time expectancy for that is about 8 months. And at 8 months, the FDA will certify the site for manufacturing. And then we'll spend another 4 months developing 3 months developing the IND approximately. And then we'll submit it for IND to the FDA. And that will require a lot of other information from principal investigators and all that. And then once it is submitted for IND, we expect about, like I told before, between 3 and 6 months to get the IND approved. There's a lot of back and forth with the FDA. And then we go to the IRB in various in OHSU and Ghent, and the IRB will have questions before they approve the clinical trial. Since it's a very small trial of about 10 patients, I don't think that there would be many issues. It's an open label study, so there's no question of blinding and all that. And so it will be a relatively easy process. It will be mainly geared towards safety of these patients, because we are going to be taking them off antiretrovirals. And so the issues will come with consent of the patients. The consent will have to be very detailed so that patients are protected. And then monitoring of these patients so they don't fall sick. That because we're going to take them off the drugs to give the vaccine. So if we take them off the drugs and give the vaccine, how often are we going to monitor them to restart the drugs if something goes wrong? And that is where the IRB will be focused. I don't think there should be any other issues with the trial. So the monitoring and how long we'll follow them up after the trial. Those are usual IRB concerns. So it's not going to be that much more different than trying a new drug or something like that. If that addresses the question. And then once we have got the IRB approval, the next timeline is the actual clinical trial. And because we are recruiting 10 patients only, and maybe in 2 different places, in Ghent and in OHSU, we could probably recruit them within a month or 6 weeks. If we are looking for 300 patients, it's going to take longer. So the recruitment time will be relatively short. I don't know what it is right now. Until we have the actual protocol, it will be difficult to tell what the recruitment time will be. But I estimate it to be about 4 to 6 weeks to get that few patients. And then we do the actual clinical trial. So we have to take them off the drugs. And after they're off, and we will not select any patients that are on long-acting antiretrovirals. But then we'll be waiting a long, long time before we can inject them. So we are going to take patients that are on short-acting antiretrovirals. We'll take them off the antiretrovirals, and within 10 to 15 days, we'll inject them with the first dose of the vaccine, keep monitoring them. 1 month later, we'll give them the second dose of the vaccine, and then keep monitoring them for 24 weeks thereafter. So about 8 months total would be the clinical trial. And so that is the timeline. So we are anticipating between 24 to 30 months, we'll have the results of the phase 1 trial.
Mhm. Okay. We have one question that looks pretty far afield, and it is, what are these strategic plans for the company beyond the successful launch of IMTV-014? Are there any therapies in the pipeline?
Yes, but I don't want to muddy the waters. The immunological approach that we are using and the knowledge can be applied in oncology. It can be used in Alzheimer's disease. In new research shows that Alzheimer's is a disease of the immune system, and so there are many pathways that the company can take long term. But right now we want to focus on this completely. This is very important for the entire world, that we research this and bring it out into the market, and not be deviated for having a pipeline just for the sake of having a pipeline.
Okay. And I know that you've touched briefly on this subject during the presentation. One question that we have, can you elaborate on the patent strategy, particularly in regards to future patents?
Yes. Our original patent was obtained on a process that we no longer use for various reasons, because that particular process under which we got the patents would not be approved by the FDA in today's world. So we used an entirely different process for the animal studies. Starting in 2010, we developed a new process, and this is the technology we are acquiring. It was developed by the prior company, and we are acquiring this technology from the investors of the prior company. And so there are multiple steps that go into making the vaccine, and I might venture a little bit into the forest here, but making a vaccine is very much like farming. So you got soil and you got a seed, and you mix the soil with the seed, and then the plants grow, and then you make more seed. So it's very similar to that. So you have what are called cell lines, and the cell lines are then used to grow the cells first, and then we have the main virus, the prototype virus that we have for the vaccine, and we drop it into the cells, that is the seed, and that virus infects the cells and creates more virus, and when we harvest it. And in this process, we found that the NEF-deleted HIV grows 40% slower, 40 times slower, not 40%, 40 times slower than the wild type HIV. And so we are, in order to improve the yields in a short period of time, creating a robust process, we used two different kinds of cell lines, mixing them together, and that has not been done before. And again, it's all proprietary information, so I can't really give many details on that. So that is one patent. The second is how we seeded them. So instead of seeding them like a conventional approach, we seeded them a little bit differently. And so that difference in seeding is another patent. Then to remove the debris from it, we used a very specialized and custom-made filtration process. That is patent number 3. Then it goes into what is called tangential flow filtration for further purification. And the tangential flow filtration creates its own unique challenges. So we resolved them by developing a custom apparatus. So that is fourth patent. I mean, I could just elaborate more and more and more, but we have about 50 different things that we did uniquely. and we didn't apply for that because they both meet the patent standards of 35 CFR 102 and 35 CFR 103, which is novelty and an obviousness. For utility, we have to wait until we do the clinical trial. So that is 35 CFR 112.1. We will meet that once we finish the clinical trials, and then we'll be able to apply on all of these patents.
Okay. One question that we have. So the acquisition is part of the fundraising strategy?
Yes.
Okay.
Yeah, and it will be completely acquired, not licensed. So it will be Immgenuity's. It is Immgenuity's technology. So there is no royalty or any payment to be made to the prior investors.
Okay. Okay, and I think that those are all the questions that we have.
Okay.
So, if there are any closing remarks that you would want to add?
Yes, I'll nominate David Miller to give the closing remarks. David?
Yeah.
So closing remarks, please.
Yep. Throughout the past year, we've been able to attend and present at dozens of conferences. We've got 63 million people worldwide infected with HIV. That's predictive epidemiological analysis. Because of the war in the Ukraine and the fact that the Ukraine had the highest rate of HIV in all of Europe, that number is now being accentuated. We're seeing a whole new rise in HIV infection throughout Europe because of the Ukrainian diaspora. The circumstances with HIV aren't just about HIV. It's about retrovirology and virology itself. We've been able to produce significant scientific agreement and recruit a unparalleled scientific advisory board, which affords us, to all intents and purposes, a ironclad opportunity to enter into publicly sponsored clinical research networks, which means we're not paying for these studies. After phase 1, we're going to be petitioning the NIH for the research. We're also talking about remission, which means that we're talking about presenting a cost-benefit analysis. If this works, which we have more than enough rationale to contend with that this is going to be a viable, efficacious intervention, that shows a novel approach to virology. But there's one important point beyond what the potential is and what we've done. We all just went through the biggest experiment in therapeutic vaccination in human history, COVID-19. Both of the technologies, BioNTech and Moderna, developed from HIV vaccine studies. That's what they were doing before COVID came about. And more people got a therapeutic vaccine than any other time in human history. It's opened up a door. It's reaffirmed therapeutic vaccination. Therapeutic vaccines have been tried in HIV before, and they failed. And they failed because they didn't have the audacious premise that Dr. Apte has prosecuted with the Scientific Advisory Board to afford and allow an address of NEF, which is the culprit that's widely regarded in all the peer-reviewed literature to be the cause of the disruption of immune signaling. So the scientific and financial opportunities here are profound. We fast-tracked all the COVID-19 therapeutic vaccines, and we have all reasons to believe that we'll be subject to fast-track designation as well. There's a lot of investors sitting on the sidelines now, wringing their hands and piling up the Kleenex, wiping their tears away when they didn't invest in Moderna when they were $4 a share because they weren't buying the HIV vaccine story. COVID came around and it changed everything for the biotech arena, for infectious disease. There's a lot more to learn and to understand about the prosecution of this scientific premise in an effective manner so that commercial opportunities become imminent. And I invite anyone to start wrapping their mind around what kind of a scientific achievement this is going to provoke and what the returns on investment are, because this type of innovation brings forward rewards. That was my close.
Thank you, David. And thank you, everyone else. Thank you, Gabriel, for the opportunity. And thank you for the investors that are still with us. And thank you for the questions. Thank you for asking the easy questions.
Yes.
And I look forward to having harder questions in one-on-one contacts with any interested parties.
Yes, as I announced at the very beginning, a copy of this recording will be shared amongst everyone that participated. That being said, again, thank you so much for attending, and hopefully we'll talk again soon with everyone's, as Satish said, during the harder one-on-ones.
Thank you.
Take care,
everyone. Have a good evening, good morning, good day.
Thank you.
Take care. Bye.
Thank you, everyone.
Thank you. Bye-bye.
Bye.